robot would say. We're live. Are you a robot? And if you were a robot, would you say yes or no? What would your choice be? That's an interesting thought question. But this is a podcast about science, and we're going to start the show in just a moment here. I just want to let you know that if you're not yet subscribed, make sure you subscribe to us wherever you're watching us. You can find us YouTube, Facebook, Twitch, and click that notification and that like button and be notified all the time for when we're going live. Additionally, this live show will be edited for the final podcast, because that's what we do. So if you like edited things, subscribe to the podcast too. Are we ready to start this show? We're ready to start a show. Yay! We turn things on and make it go. And we will begin a show in a little under 3, 2, 1. This is TWIS This Week in Science, episode number 813, recorded on Wednesday, February 24th, 2021. How to choose your roach. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with dating, gliding, and fighting. But first,
disclaimer. Planet Earth is in big trouble. Pandemics, global warming-induced weather patterns, the lack of sustainable solutions capable of overcoming human laziness. The end is not in sight, but it is a good sight closer than it was just a few years ago. And yes, some humans are working really hard to ensure that changes are made to keep this planet habitable. Meanwhile, Mars. It's not ready to live on, not just yet. But the worse it gets here on Earth, and the faster we need to come up with solutions to survive here, the better Mars looks. It won't be for everyone, but for anyone that does go there, one thing they will need to bring with them is This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
and a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back with all the science that we can fit into this show, which is usually quite a lot, and I'm very excited to have pried myself off of a very warm, comfortable couch to come talk about science this evening. Yes, I am. I have stories tonight I've got some stories about artificial intelligence and video games. I also have a story about magnets, magnetic poles flipping. I've got a brainy story and a slimy story for my section in the end of the show. Fun stuff. What do you have, Justin?
That story. I saw that one too. I've got dating sites with Aboriginal wasps, how to win at cockroach fighting, why taxes and red tape are a good thing, and mushroom soup.
I love mushroom soup.
Yeah, that's good stuff.
It's delicious.
Yum.
All right, Blair, what is in the animal corner?
I wish mushroom soup. I have
about it.
cloned ferrets and monkeys watching TV. And then I also have a fun story about an underwater glider.
Underwater glider.
Yeah.
Like a fish.
Just imagine a paper airplane underwater.
For
For science.
Great. For science.
Yeah.
For science. All right, I will do that. Everyone, as we jump into the show, I would like to remind you that if you have not yet subscribed to TWIS, you can do so on YouTube, Facebook, and Twitch. You can also find us all places that podcasts are found. Look for This Week in Science, and our website is twis.org. Yeah.
That's it. That's where it's been. It's been there for a while.
It's been there for a really long time. It's still there. It's just
Waiting for you.
the website that keeps on being a website. Webbing. That's right. As long as I keep paying the bills for it. All right. Justin, your disclaimer today reminding us of going to Mars and
Wouldn't that be nice to go there one day?
the robots that we are sending there in our stead. Last week, it was the night before the Perseverance landing. And this week, we are about 7 days, 6.5 days since a successful landing of Perseverance on the surface of Mars. Woo!
Yeah, it was pretty exciting.
It was very exciting. And all right, the 7 minutes of terror, we, for the first time as of Monday, have a video from NASA that brings together footage from off-the-shelf cameras. They decided, finally, how many years have we been saying, put a camera on it and send images back, right? Let's see some real video of what actually happened. Well, off-the-shelf camera, two or three of them actually, an off-the-shelf microphone, and they sent back a video that is outstanding of the 7 minutes of terror. For the first time, we have video of humans, well, our robots, landing on another planet.
Can we watch it? Or
And it's
no?
outstanding. Oh, yeah, we can watch it right now. I might clip it out of the...
Just a second.
It's a 3-minute long video, so I think it's a bit much. But I do also want to share with everybody before, maybe we can watch it in the after show.
Okay.
I think that would be good. But I did want to share with everybody the first audio from another planet. We have a microphone on Perseverance, and it was able to record, not during landing, because that was very violent and really loud and probably didn't get very good audio. It didn't work. But after landing, just shortly after landing, this was sent back.
This week in science.
And what you hear right now...
Yeah.
Yes.
Wait. Is that our theme song playing on Mars? I think I heard it.
Sounds like it. I think it's carried on the breezes of Mars. Yes. But that sound is the sound of Perseverance itself, the electronic sounds that the rover was making, and the sounds of the wind. And they have taken out the sounds of the rover. And that's the sound of a gentle breeze.
On a microphone. Yeah.
On a microphone.
You can't mic wind. It's not a thing that you can really do successfully and have it sound like anything interesting. I'm sorry.
It sounds like...
No, I think that's very cool.
I'm not saying it's not cool. I'm just saying, like, somebody blowing into a microphone or wind on a microphone. Ooh, like, it's just not the... You know what I mean? It just doesn't really... Like, hearing the drill operate, maybe, when it's doing the drilling, that might be cool. I don't know.
Or if there are other sounds, because perhaps it'll be capturing sound when there's a Mars quake. Perhaps it'll be capturing sound. Who knows? I mean, is there a dust storm going to get going at some point? Will the sound of the wind increase? What kind of information can we get from being able to hear?
I don't know. Blair does not seem impressed. She's over there yawning.
Okay.
I thought when we were watching it, we were watching it sort of live. And you were talking about that 7 minutes of terror. Like, we're gonna lose communication, can't tell what's going on with the thing. It's gonna hand off to another thing. But it's an 11-minute delay. So even though everybody's waiting for this 7 minutes, it has already happened. It had already taken place because the delay was 11 minutes at that time.
Yeah, and I'm seeing in the chat room, terror is a strong word, says Fada. I don't think so for the individuals who spent the last, 8 years on this lander?
Right. And even
They
though
literally
they
had one
were
job. They literally had one job for 8 years, is to make sure that at the end of this 8 years, nothing goes wrong.
I'm not sure about that number. You keep repeating my 8 years, but I'm not 100% sure about that.
Well, we have images from the surface of the surface of Mars, from the area, from the Mars Reconnaissance Orbiter as well, showing the variety of objects that have been left on the surface of Mars. We have a heat shield well off to the east of the landing site. We have the landing site itself. There is a location where the sky crane descent craft actually flew off to and potentially exploded. There's a picture that's been released of a giant cloud. We don't know whether that is a dust cloud from landing or if it landed and crashed and exploded. And we don't know that for sure. There's also another shielding component that's off on another area within this landing site of Jericho crater. And knowing it's all there, I think, is very interesting. This human debris, our leftovers, signs of humanity on a distant planet. Even though it's within our solar system, it's a distant planet. And this is humanity reaching out. And science and engineering are taking us there. And it's just incredible.
And as soon as we get there, we start littering. Come on, guys. Can we clean it up a little?
So also, I checked
Not tell them exactly who we are on the first day.
Just so I don't get a million tweets about this. So it's been 9 years, I guess, since Curiosity landed, which is where that number was in my brain. But Perseverance has been going for about 11 years, the project.
Yes. Well, it was first, I believe it was 2013 or 2012 that it was proposed, right after Curiosity landed. They were like, let's do more of this. And they said, let's send this lander. And there was funding, and the Mars 2020 mission was off to the races. And they rapidly developed this.
I don't know where this 11 years.
Well, that can be right too. They can already have been working on the lander and its capabilities. Yeah. And then said, hey, everybody was happy that we landed successfully. We've got another one we can send.
Bigger. It'll be bigger and better. And it'll take little regolith poops. Anyway, if this is the first time you've heard that term from TWIS, you haven't listened to enough episodes.
No.
Anyway, I am very, very excited for NASA. I am excited for all the individuals who have been spending a good part of their career in developing this mission. Now the scientists who are going to get to run the science part of the mission. I'm looking forward to all of the results that come from this, and it's going to be so exciting moving forward. I just wrote, after Curiosity, there were all these bits of information that came out. Insight brought us bits of information, and this will just give us even more. And
I know they're far away from each other, but can they go to each other and interact? And
so they are actually social distancing.
Yeah.
They are.
Like we are.
Yeah. I just think it would be cool to get pictures of a rover on Mars, right?
No, we didn't go to Mars to take selfies. Everybody needs to stop with the selfies.
I don't know.
Mars.
I think it'd be kind of cool.
It would be, actually. It'd be pretty cool.
Yeah.
Yeah, it would be cool. But we are placing these rovers in different places around the Red Planet so that we can answer a variety of scientific questions and spread our resources around. If we only looked at one spot, it had all the rovers looking at each other. Our findings would be more
later. Like when the place is riddled with robots. I just want pictures of robots on Mars. That'd be very cool.
Yes.
I mean, when little Ingenuity's up and going, he could go over and take some photos.
Yes, and thanks for mentioning Ingenuity. Ingenuity phoned home and everything is good to go. That little helicopter will be soon into the Martian skies, which
I can't wait
exciting.
for that. I'm very excited about that.
Yes, and Michael Gibson is saying also, as we were talking about the different locations, why they chose Jericho Crater is because there are the hints of an ancient waterway there, an ancient lake bed, and they landed, as Michael says, in what is suspected to be a river delta, which is very exciting. Yes, so many fun characteristics to check out and learn about, and we get to learn along with them. So thank goodness we didn't have to worry. We didn't have to be talking about a blown up, exploded Perseverance, and that we can be excited moving forward. But Justin, what's your next story?
Let's see. There is a 2-meter long painting in Western Australia's Kimberley region, which has been identified as Australia's oldest intact rock painting.
Ooh.
And they were able to date it using radiocarbon dating on 27 mud wasp nests that they were collected from. Some of them, so this painting's on the rock wall, but some of it is also over the wasp nests, which has created a very convenient way for them to radiocarbon date the wasp nests that got stuck behind the painting compared to the ones that ended up on top of it on this sort of the roof of this cave. This makes the painting Australia's oldest known in situ painting, says postdoctoral researcher Dr. Damien Finch, who pioneered the new radiocarbon dating technique. This is 600 generations ago. This painting was put on the roof, and they say it gives you an insight into what they were thinking. The painting is of a kangaroo. Maybe not super surprising. But what's sort of interesting is there is a shift to the really old artwork in the cave, because it persists over generations with more and more work showing up later and later and later generations. But the oldest artwork is life-size. If you're gonna make a kangaroo painting, you make it the size of a kangaroo, obviously. And then at some point they realized, you know what? We could make the really small depiction of a kangaroo, and we know what it means. I
just imagine trying to like doodle in the margins of something in life size.
But it's sort of funny. Like it's almost like this was the first big renaissance in the art movement. You know, it's not just when they've got the depth perception of within the painting, or they've gone to some other abstract technique. It's like, you know what? We don't have to render everything in full size. We can scale it down once in a while. Yeah. So it also helped in dating some of the other art, because a lot of the works are sort of painted over previous works. They sort of overlap in some areas. So you've got these wasp nests behind. They can be radiocarbon dated. And then you've got maybe paintings that are overlapping that one. You sort of know what order they came in after. And you have other wasp nests that are in there, and those can get radiocarbon. So they got it back to between 17,500 and 17,100 years ago. Dr. Sven Oosman from the University of Western Australia School of Social Sciences is one of the project's investigators. So the rock paintings would unlock further understanding of some cultural history, and points out something interesting. This iconic kangaroo image is visually similar to rock paintings from islands in Southeast Asia dated to more than 40,000 years ago, suggesting, to him at least so far, a potential cultural link between the 40,000-year-old paintings and the 17,000-year-old ones. So and he says it might mean that there's even older rock art that they haven't found yet in Australia.
Right. So he's saying that this is like a ancestral lineage of peoples who have we could potentially tell that because of characteristics of the art. I'm just thinking, don't you think your art would evolve a bit over like 20,000 years. Maybe change a few details here and there. Use some new material.
Yeah. But if it doesn't, and that might be kind of the thing, like, we looked at this. You look at the stone tools that were being used. Some stone tools didn't change for 1 million years.
Yeah.
They got passed down. You learn it. There wasn't a big cachet on innovation. Apparently, people weren't totally getting impressed with the new way to make a kangaroo cave art. People look at that as like, that's not how we do kangaroos. Where are you from? You're not part of tribe if you can't make kangaroo like we make kangaroo. Okay, fine. I'll go put back. I'll fix it. I'll make it life size. It's not that hard. I can do it. But there wasn't,
Make it bigger.
It must have also been like, if you got too creative, I think in the ancient times, tried eat that berry for the first time that you hadn't seen anybody eat. Tried to take on that prey that no one else seemed like they ever hunted before. Like, right. I think it usually ended badly. Mm-hmm. I'm just going to, I'm sleeping outside this winter. I'm not even going in the cave. I'm done with the cave. It's too stuffy in there. Never saw him again. Well, until the following spring.
Yeah.
It must have been bad trying to be too creative back then. Anyway.
Yeah. Don't be so creative. Just survive.
Just survive. It was just hard enough just surviving, playing by the rules.
Yeah. Blair, what about gliding through life?
Oh, yeah. I just want to glide. So if I was trying to send a robot into the ocean to survey areas of the ocean, what are my limiting factors for that robot?
It has to be waterproof.
Fuel?
It has to be waterproof for sure.
Energy?
How long is it going to last?
Pressure might be if it goes too deep.
Sure. Mm-hmm. And then also, if it's super loud and has a giant propeller on it, you think you're going to see many animals?
Drive off everything. And it's dark down there, too. Mm-hmm. Yeah.
So, this is a study from Purdue University. This is actually Nina Mahmoodian, Associate Professor of Mechanical Engineering. She actually started this project in 2012. Everything's 2012 today. At Michigan Technological University. And she has since developed, tested, peer-reviewed, now sent out, Ruffy, Roughie, as in, isn't that how you spell orange, Ruffy? Is the research-oriented underwater glider for hands-on investigative engineering.
Aha.
So now you're just making things fit acronyms. But anyway, ultimately, what's cool is it's highly maneuverable. It's low cost. And it is a glider, which means there are no external moving parts. It looks like an underwater paper airplane, as best I could describe it. So they also can last a lot longer. So a robot, an ROV can just last a few hours a lot of the time. This guy can go for weeks to months between charges because there's less moving pieces. So there's a lot less to charge, essentially. And so that can be deployed for days to weeks to months. And it doesn't have those moving pieces, so it's not going to scare away animals. It's shaped like a torpedo with a static rear wing. So for those of you listening, you can look it up. There's some really cool videos. There's a YouTube video that shows exactly how it works. But basically what it does is it pumps water into ballast tanks to change its buoyancy, which provides the glide path angle. Is it going up or down? And then to control its pitch, kind of which direction it's pointed in, they shift weight forward and backward. And then they also steer with inner components mounted on a rail that rotates. So you can imagine, you know what I was thinking about was, this is like me as a 90s kid. They had those pens that would vibrate, that were like squiggly pens. Do you know what I'm talking about?
Mm-hmm.
Yes. So the way that those worked was there was a weight that was off center that was rotated around with a gear. So I picture it like that. So basically they're able to move this weight around to be able to adjust where it's being dragged towards. So everything is on this pole inside of the torpedo in just this very compact space. So you'd be able to make different shapes, different sizes, and apply this to other types of gliders as well. It also has a much better turning radius. So previous gliders have about a 33-foot turning radius at best, but this guy has a 10-foot turning radius. So much
it is
easier.
It's a compact design itself. So it being so small, it's going to have a smaller turning radius.
Yeah. Absolutely, yeah. And so it was such a compact turning radius, and it was so much more maneuverable than previous gliders that they could actually test it in the swimming pool, which I thought was very cool. So they've used it in the swimming pool, and then they've since also sent it into ponds and lakes to measure algal blooms, but they hope to send it out much farther, like to shipwrecks, underwater.
To shipwrecks.
Like yeah, very
cool. Mm-hmm. Yeah. Yeah, I wonder if they could develop designs that are not just for short-term use, but that could maybe like recharge and go out for longer periods of time and be more autonomous. But
put some solar panels on it, and when it's powering down, just have it surface,
and then collect, and then go back down.
Yes.
Yeah, exactly. Yeah. You're following me.
Yeah. See you in a couple years, buddy.
And now I'm following you with a story about artificial intelligence exploring video games. Yes, I did say artificial intelligence exploring video games. Researchers have been playing with artificial intelligence algorithms, a group of them that they are calling Go Explore. And Go Explore, they set on Atari games, including Montezuma's Revenge and Pitfall. And
I like the Pitfall game.
Yeah, Pitfall was super fun. Okay, and so when you are playing the game as you're going along, you have a memory of where you've been, what you've seen, kind of what areas you've tracked through, and you know what the goal is. You always have the goal in mind, but you can keep from getting distracted. You can keep from getting sidelined. You can kind of figure your way around things. And so the question is, how do researchers start teaching artificial intelligences, these algorithms, to do the same thing? So these generally intelligent agents, these algorithms, sometimes they just get unmotivated. They're not properly motivated to actually find the goal.
Some kind of proper carrot for the artificial intelligence.
Are they just getting bored? Are they getting bored with the video
reinforcement or
it's
negative reinforcement? I don't know what works on robots.
Kind of. So what they say is that say you have a house, a map of a house, and the house has a refrigerator in it, and the AI is supposed to find the refrigerator. Well, if you're trying to get them to the refrigerator, what they say is that someone might provide a reward only when the refrigerator is reached. But since there's only one refrigerator in the entire house, there are a lot of actions that are required to get to the refrigerator. And so you kind of have to have mini rewards all along the way to keep the motivation up so that keeps going. And so they write also a denser reward. For example, the Euclidean distance to the refrigerator can be deceptive. Naively following the reward function may lead the robot into a dead end and can also produce unintended and potentially unsafe behavior. For example,
trying to open the cat.
Yes.
The refrigerator.
Yes. Or, falling down stairs because they didn't properly, avoid them, for example. So you have to figure out how to space the rewards so that there are denser mini rewards. And then also you have to keep the algorithm from getting detached, which is that it won't that it stops returning to places. And maybe those places are important to ending up at the end goal, but because they've already visited them, it's like the Roomba that never goes to like the one corner of your room and it's full of all the hairballs. The room, you're like, why wouldn't you go there, Roomba? And the Roomba's like, meh, I'm detached. Algorithms. Algorithms also get derailed. So that means instead of going back to like a save point or a safe space that they've already been before exploring back to that other space. So they, instead of coming back to a place repeatedly, they'll get derailed and end up going off and be like, I think I'm going this way now. So there are lots of issues to deal with, and the researchers cataloged, they changed a lot of these things with the Go Explore, because they gave Go Explore algorithms the ability to have lots of those save points, so that they might, the algorithm might start at one point and then move out from it, but then make a new save point after it's covered a certain amount of space and be able to move forward. Anyway, long story short, because of the adjustments they made to these factors within the Go Explore algorithms, they were able to get these AI to by far beat the best human player on Montezuma's Revenge. Like, it just blew humanity out of the water when it comes to playing Atari games. Just got the super high score ever scored. Yeah, it's superhuman, the score, its performance on this game. And in the process, because of what they've learned, they're hoping that they will be able to not just have AIs show us, how much better they are at video games, but actually help us in things like molecule discovery or drug discovery, or any of these very large data sets that have to be explored and sites remembered and paths taken. And maybe this kind of artificial intelligence algorithm can help us in our data and intelligence capturing moving forward.
Yeah.
Go explore!
But it's just like you said, it could also be used to dominate online video gaming, and
it can as well.
No humans are allowed to play anymore. Or anything else that people want artificial intelligence to do.
Yeah.
Anything. I mean, that's we talk about it like, we could find a cure to a disease because we're doing a science show. If you're in the financial speculation sector, you might go, now would be a good time to short sell GameStop because the robot told me to. And then somebody else's robot's going to be like, hey, I got an idea. Somebody just short sell GameStop. But it could happen across the financial markets instantaneously, back and forth. War of the AI is probably going to be the most impactful outcome of this, I think.
Right. So, I mean, at some point, it's going to be AIs battling AIs.
Coding, hacking, AI hacking computers. No digital data safe anymore. Everybody goes back to paper. Everybody has to go back to paper because the AI can hack everything in the future. There's going to be downsides. I know you don't want to hear about them, but I'm just
You mean
terrified.
There will be pitfalls?
Oh, I love that game, though. The jumping over the elevator. It was like an archeologist game, I think. And he's went over there and jumped over there. And
always fell in the pit. Always.
Oh, the pit. The gators get you once in a while. It's going to happen. It's all in a day's pitfalling.
Pits, quicksand, rolling logs, fire, rattlesnakes, scorpions, and crocodiles. Pitfall Harry must jump over or otherwise avoid these obstacles by climbing, running, or swinging on vines.
She had to Google it. She had no idea what we were talking about.
I played Atari games, but I wasn't aware of quite a large library of them, let's say.
I think that was one of the rare Atari home video game, and you could play it at the arcades, like the early arcades. I think that was one of them.
Yeah, it was. That's where I always fell into the pits. Justin, tell me, if we're not going to the arcade, we're going to the cockroach pit fights, right? So
doesn't happen. It doesn't happen often. But like anyone else, it happens to all of us occasionally. You're walking down the back alley path of some part of town that has seen better days. You follow the muffled sounds of people cheering, the faint whiff of cultivated smoke in the air, down some below street level stairs through an unmarked metal door, and suddenly you find yourself where the action is hissing. Cockroach fights! You want to place your bet with the three-fingered man in the orange visor, but how do you know which roach to ride? Science to the rescue! Turns out, not all cockroaches are equal. Some are super athletes with larger respiratory systems and are more likely to win in physical mating battles. So this is research published in the journal Animal Behavior, led by Dr. Sophie Mauls of Anglia Ruskin University. She studied aggressive interactions between male wide-horned hissing cockroaches, Gromphador hina oblongata. The animal contests are usually won by the larger opponent in physical fighting, and that's pretty much the same across all of the animal kingdom. And they also can sort of size each other up and go, yeah, I'm not going to fight. You win, because I can tell you're bigger. So what they did was they took a bunch of same-sized cockroaches and they put them up against each other to see what would happen. And they studied the winners of those battles, or those who were rated most dominant, whether they won the battle or sometimes maybe even just scared the other one away. CT scan of all the cockroaches was then carried out and allowed them to see the whole body, including the size of the respiratory system. And that was when they realized, hey, all the winning cockroaches have bigger respiratory systems. So anyway.
So they look the same from the outside.
Look the same from the outside.
But on the inside, they're very
totally
different.
So they're getting more oxygen. They get more oxygen. They can fuel their little cockroach muscles to twitch faster.
Don't fight,
stronger.
buddies.
But yeah, so if you go to a cockroach fight, bet on the cockroach who isn't smoking.
With the biggest lungs.
The one with the biggest lungs. So, yeah.
Blair, you look concerned.
That's just that's a wild thing to do in the name of science. I get it. I just wasn't expecting to see a cockroach cage match today. That was not on my radar.
I mean, really, both cockroaches they come out just fine in the end.
So
it's fine. It's not like they ate their wings off, like your story last week.
Fair, yeah, but that was consensual.
Could be way worse.
It could be worse.
What? So the worst that happens typically in these is one cockroach gets flipped over on its back and then does this for a little while. Ah!
It was just especially with the red lighting.
Oh no, that's actually from one of the cage matches. That's one of the cage matches. The red light cage matches are to the death. That was different. What you were witnessing. Yeah, you just went to the most extreme version of the sport right away.
Oh no. Oh my goodness. All right. I have another story that doesn't have to do with cockroaches. Well, kind of does. It has to do with running in underground and hiding in the cracks until it's safe to come out. So I guess that it has to do with cockroaches, except it has to do with all life on Earth because researchers in Australia, they looked at a really, really, really old tree. So these researchers were looking at a tree that it's the New Zealand kauri tree, and it's one of the most ancient types of trees in the world, and it gets preserved. There are trees that have been preserved by the swamps and the bogs, and these trees can be dated way, 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 way back. And so these researchers dated the trees back to the Lachamps excursion, which is about 41 to 42,000 years ago, about at the last time that the Earth's magnetic fields flipped. So the researchers looked at cross sections from 4 of these old trees going back to about 42,000 years, and they analyzed them for carbon-14. Now, why did they look for carbon-14? Well, the reason is that when the magnetic field of the Earth weakens, more cosmic rays make their way through the atmosphere, through the surface of the Earth. And as they make their way through the atmosphere and they come into contact with carbon, they turn it into, they cause the reactions that lead to the production of more carbon-14. So weaker magnetic field, more cosmic rays, more carbon-14. And so they thought, we've got these old trees. Let's look at them and see if we can find a signature of increased carbon-14 dating back to the time of that last magnetic pole flip. And that is what they found. They found increased levels of carbon-14 in the trees going back to about 41,000 years ago. And this supports a hypothesis that has come out a few times over the years that periods of magnetic pole flips are potentially very dangerous to life on Earth because of the increase in cosmic rays, decrease in ozone. So there's more radiation solar radiation making its way through the atmosphere to the surface of the Earth. And they've got all sorts of things that back this up that are really interesting, which tie in a little bit to the paintings on the cave wall story that you were talking about, Justin, because they suggest that there is an increase in cave paintings about 42,000 years ago because people went underground to survive. That's the idea. And so they went underground and they're like, hey, I'm bored. Let's paint. We don't know if that's true, but there are cave paintings back then. But people have always lived in caves, so I think that's
off and on,
tying
just
it together.
Depending on the weather more than anything. Yeah.
And so if you go outside, you get burned. I mean, it hurts when I go outside. You'll go outside, you hunt, and then go back underground again.
But 40,000 years ago, no, that's too long ago. But that's interesting. That's interesting. It does seem like when the major die-offs that I saw was being attributed to this, they kept pointing to large fauna.
Yeah.
That largely disappeared in the overlap of mankind.
Yes.
Also. So that's
it could be mankind, our influence, but it could also be that kind of thing that the environment changed. The climate changed. There was an increase in solar radiation, a change in the atmosphere that led to more atmospheric hydrogen, more nitrogen oxides, less ozone because it's getting consumed by those things. And because there's less shielding from radiation,
but
you'd see a lot of changes in weather patterns. They suggest that there could have been massive cooling to the planet as a result of the magnetic field shift. But lots of things tying back, and it's just very interesting that they have this bit of evidence from these very old trees in New Zealand. Yeah. So, I mean, it's also interesting because we are potentially going through the beginning of a magnetic pole shift right now. So what's gonna happen? We all live inside anyway, so it doesn't matter.
Wait, how often are these things?
It's every 24,000 years?
No.
Something like that.
If it's that often, then
42. No, I think it is every 42,000 years. I think I got yes, it's about every 42,000 years. It's a pretty consistent amount of time.
So it's any day now.
Pole shifts. Yes.
Any minute.
Any minute, any second. I mean, we've been talking about this for probably
that's how we solve
20
global
years.
Warming. How do you make one happen? We need a cooling.
Right.
This is our shot.
That's our one shot. That's right. Magnetic field flip. Let's let it happen and see what happens.
We already live in caves. We're there.
I know. Gord, yes, we all live inside. That was a shout out to you. No, I'm kidding. Yes, the cave paintings, John Hogan saying the cave paintings have major celestial events associated with them. Sometimes they do. Yeah, not all of them, but some of them do. And so, yeah, how much were they staying? Celestial, you're going out at night, not during the day when the sun is hot and burning. I don't know. There's lots of interesting pieces here. I don't think anyone has put the entire puzzle together yet at all. And there are still massive questions outstanding. But it's one of those unknowns. Life has obviously survived for millions, billions of years in one form or another. Humanity has survived for over 200,000 years. Humans in one form, primates, have survived for much, much longer. So it's not as though this is something to be really concerned about, but really, what should we be concerned about? We don't know. I think it's very interesting. I am fascinated by this question, and I hope that you are too. This is This Week in Science. If you just tuned in, welcome to the show. Thank you for joining us. If you've been here all along, we are so glad you're here once again. Oh, hey, do you need a TWIS shirt or some kind of a mouse pad or a mug or something? I don't know, maybe you need a new hat to shade your face from all that solar radiation. Head over to TWIS.org and click on our Zazzle link to find a bunch of TWIS merchandise that will help support the show. We appreciate your support. All right, everybody. Back to the show. It's time for a COVID update.
Oh, it's that time, huh?
Yes. Well, I just want you to know that according to the CDC, vaccines are just doing awesome. I mean, we're not getting them out as fast as we would like, but we really are getting them out there. And that's just awesome. I think props to people getting vaccines, to getting vaccinated. It's all helping to push this pandemic a little further away every day. 13.8 million doses of vaccine were administered in let's see, where was this that the CDC reported on? Yes, the first month of COVID vaccine safety monitoring. So this time period is December 14th to January 13th. And we're now end of February, so you know that there's much more adding on to this. But in that first month, the data from that, 13.8 million doses of vaccine were administered, 6,994 post-vaccination adverse events were reported in the VAERS, the Vaccine Adverse Event Reporting System. And the most common symptoms were headache, about 22%, fatigue, 16 or so percent, and dizziness, again, 16.5%. Anaphylactic reactions were reported in approximately 4.5 out of every million vaccinations, which is similar to the rate that's expected for inactivated seasonal flu vaccines. So it's to be expected and not anything out of the ordinary, which is great to see. Adverse events were more likely to be reported after an individual's second dose than their first dose. And additionally, within the data, there were 100, let's see if I can get the exact number. There were 113 deaths reported in the adverse reaction database. However, all of the information has been looked at, and they have confirmed that it does not seem as if any of those were actually as a result of receiving the vaccine, that all of them were just other reasons, but were reported because they were within the 2 months of getting the vaccine itself. So
I think it's also important to note these numbers of the adverse reactions: 22% headache, 16% fatigue. So if you talk to friends and family who've gotten the vaccine and you look at social media and stuff, you would think that number was like 95%.
Yeah.
But part of the reason is that the people talking about it are the ones who had bad reactions. So first of all, you're going to hear more out of the people having a bad time than the people who didn't. But on top of that, our brain is programmed to remember better bad stuff.
Right.
So I think that's my, I don't have any data on why it seems that way, but based on our understanding of bias and how reporting can be misleading, I'm pretty confident that that's what's going on here, because this number hasn't really increased. But
right.
It really feels like everybody's saying like, oh my God, I got a fever and a headache from the second dose. But that can't be true, because that's not what the data says. I think it really is a reporting problem.
Yeah. I would think I think that as well. I mean, it's not everybody who's having these problems. It's usually the case. We have a negativity bias.
Yeah, exactly. Yeah.
Right. Yeah. So it's very exciting. Vaccines are rolling out, and that is extremely positive. And additionally, researchers are looking into ways that we can potentially treat this, to treat COVID-19 moving forward. A research team at Penn State University just reported on their investigations of the SARS-CoV-2 Spike protein, in which they did some fun maneuvering to look really closely at the spike protein itself. This is the part that binds to the ACE2 receptor. They used amide hydrogen deuterium exchange mass spectrometry, HDXMS. And so they used this to bounce hydrogen deuterium off of the molecules that make up the spike protein as it was binding to an ACE2 receptor, so that they could actually see what was happening. So this deuterium oxide, they use heavy water deuterium oxide, and this probe, they let it come in there, and it's like getting a footprint, but getting these little tiny snapshots as the components come together to see exactly what happened. The researchers say if you put the spike protein and ACE2 receptor into a solution that's made with deuterium, the surfaces and more floppy regions on both proteins will more readily exchange hydrogens for deuterium compared to their interiors. And footprints of each protein on the binding partner can be readily identified from areas where you see little deuterium and only detect normal hydrogen. They were able to determine that the spike protein and the ACE receptor is necessary for the activation of what are called furin-like proteases. And these are enzymes that are like scissors. And they come in and they snip off one of the subunits of the spike protein. But it only happens when the spike protein has bound to the ACE2 receptor. And it's like this long distance messaging that happens as soon as that binding happens. There is this long distance messaging. There's this cascade of cell signals within the cell that activate these scissors to come in and snip the subunit off. And that is what allows the S2 subunit of the spike protein to fuse with the cell membrane. If those scissors don't come around, there's no fusion. There is no viral entry into the cells. And so now that they know this and they see where this is happening.
That's brutal. So it's the cell's attempt to get rid of it turns it on.
Yeah. It's like, wham.
Oh, that's brutal.
Yeah. So this enzyme should be snipping it up, but it's not. It's helping it. It's helping it into the cells. It's opening it. Yeah. And so now that they know this process and they've seen it more exactly, what they are hoping to be able to get from this is that this cleavage might be a target for inhibiting the virus. And that, so far everybody's been like, no, just neutralize the virus. Don't let it bind to ACE2. But maybe there's another set of steps that can be used so that
if put that enzyme in every hand cleaner.
Yeah.
what was this? No, you gave us the solution. Put that enzyme, spray it in the air, put it everywhere, man. Activate the thing when it's not in a cell. Get it to open up, and it's done. That's all that it was.
Yeah.
History.
Yep. I don't know. I mean, maybe somebody's trying something like this. I don't know, but it's very interesting in light of the various mutations that are occurring and how these variants with their increased transmissibility, their increased binding to the ACE2 receptor, how are they changing and how does that affect the furin cleavage and how can we get in the middle of that to affect it? And so it's neat to know that this kind of research is going on. Blair, do you want to talk about gorillas and selfies?
Yeah, it's never a good idea to take a selfie with a gorilla. I would not suggest it. I would never do it. Nope. Ever, never, ever. But I guess there is this thing called gorilla trekking with mountain gorillas, which is part of ecotourism. And so there's a whole thing where people go and they get to see these gorillas in the wild, and so maintain a distance of 7 meters in between them and the gorillas, but they don't. So where this is really a problem is last month in January, some gorillas in San Diego Zoo got COVID, which proves that it can jump from humans to great apes, which is not a surprise. There's lots of diseases that we share with monkeys in general, with primates, and then also especially with apes, chimps, gorillas, stuff like that. So Oxford Brookes University looked at about 100 Instagram posts from gorilla trekking tourists, and they found that they were extremely close and they were not wearing masks. They were definitely close enough that they could get these gorillas sick.
Because wildlife, and they're just like, I'm in nature. I'm outdoors. Why do I have to wear a mask? They're not thinking of the gorillas as other beings.
Now, most gorillas you see in a zoo are Western lowland gorillas. They are in trouble. They are reduced numbers. They are endangered. The mountain gorilla, there's about 1,000 of them left on Earth. They are very scarce and very endangered. And so if they get COVID, it's not good. No. So this is focusing on one species, of course, but there's lots of tourism locales where people are in close quarters with primates. So if you think about great apes, that's one thing. But if you just think about primates in general, there's a lot of opportunities for COVID to jump to primates and impact wild populations. Because also, it's one thing if a zoo population gets sick, that sucks for those gorillas at San Diego, but they're not going to get gorillas at the LA Zoo or San Francisco Zoo sick. It's confined at least, right? Yeah. But if you get a wild population sick, it could get others within their troop sick. It could get other primates that live nearby them sick. It could jump to other species. There's lots of opportunity for it to get out there and get worse and also mutate, right? Yeah. Like the more species it ends up in, the more it could mutate. And, as a former zookeeper, this was not surprising for me to hear, because if we had a cold, if we had any sickness at all as a primate keeper, we had to wear full PPE. We had to wear a mask. We had to wear gloves. We had to tie our hair back, and we had to scrub in and out our boots. So there's an understanding that we're so similar genetically that you have to be so careful. So these people that are going on gorilla treks and taking selfies with wild animals in close quarters without masks on, stop.
Well, I hope that at least the people who are giving these treks, who are taking people into close quarters with these animals, will require masking. Because if these people are traveling in the first place during a pandemic, it makes me wonder whether or not they're the type to wear masks in the first place.
Can we just not do gorilla treks during a pandemic? How about that? How about just no?
How about it?
I've said before,
don't feed the monkeys during a pandemic.
If you love nature, go nowhere near it. That's the best. That's the best way. Don't try to commune with it. Don't take a selfie with it. Just stay away from it. wild be wild.
Go volunteer for a research project in the wild. This is a great thing you can do. You can do, it's called an Earthwatch trip or anything like this. So you can actually go for like it's like a flat fee. You do like a 10-day or 7-day trip. You participate in conservation research.
Yeah.
And you get to stay in this exotic locale. It's pretty cool.
I really want to do that. That's the type of vacationing I want to take. That sounds like a blast.
We should do crowdsource for the three of us to do one together.
Yeah, let's do it.
Anyway. COVID news.
Anyway,
that's what we were talking about.
Yeah, in the avoiding things vein, last story for our COVID segment, unless you all want to talk about the variants and things that are happening right now in the New York Times and Los Angeles Times stories that came out yesterday, scaring us about a new variant that we can't do anything else about except actually just staying home and wearing masks and continuing to try and reduce spread of the virus even further. Anyway, from that tangent, places you should avoid: the gym. People are breathing and huffing and puffing, and
they're not open. What?
Some
are.
Places,
some are.
They
open and then they close again, and they open. They're doing what they can. These are businesses. I understand. It's businesses that are struggling right now. I feel for, but at the same time, if you can work out at home or outside, or YouTube videos, I don't know. Gyms are not safe because of the huffing and puffing. Studies in the CDC MMR, or no, what is it called? The Mortality and whatever report. Studies from the CDC report a couple of instances of gyms, exercise facilities reporting high numbers of COVID-19 illnesses. In one, among 81 attendees of indoor high-intensity fitness classes during 1 week, 68% developed COVID-19. If your gym...
Such a high number.
It's a very high number. Like, it's really high. If your gym is not requiring masks during exercise, if your gym is not requiring physical distancing, if it is not decreasing class sizes, and if it is not making efforts to improve ventilation, you should consider a new gym. Because these are things that will help: masking, physical distance, decrease class size, improve ventilation. Oh, and by the way, same thing goes for schools. You need to follow these kinds of procedures for opening schools as well. Anyhow, can we just keep working together to complete this pandemic? That's what I would like to do. The science is here. The science working. If you have questions about COVID-19 vaccines, or about the variants that are out, please send them to me, Kirsten, at thisweekinscience.com. We'll do what we can to answer them on the show. Yeah, be good. This is This Week in Science. If you love the show, please tell a friend. That would be really helpful. Thank you for listening. Coming on back, Blair. You know what time it is?
What time is it?
It's time for Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh, the Animal Corner tonight is full of heartwarming goodness.
Oh, yay!
Yeah, balance out that COVID update we just did. So first, we have a new hope for black-footed ferrets. Now, ferrets, if you're not familiar, are the adorable weasel, if I may. But a lot of people are familiar with European ferrets. There's the ones that you see, I'll say domesticated they make questionable pets, and they're actually illegal to have as pets in California, mainly because of the black-footed ferret. So let me tell you about the black-footed ferret. They do look a lot like that domestic European ferret, but they have black feet. Done. End of story, right? No. So they live out in the plains in North America, and they eat small rodents mostly. Their number one thing that they really like to eat is prairie dogs. And around the 1980s, thank you, Kiki. In around the 1980s, we thought they were gone. We thought they were completely extinct. Chalk one up to a loss column for humans. We lost another one. But actually, in 1981, just a year later, a rancher found a small population on his property. Before that, as people did westward expansion, they were developing farmlands and homelands on these prairies that were riddled with holes from the prairie dogs. And they create these kind of like networks of tunnels underground. And it's really cool. They actually have room-based tunnels. So they'll have a bathroom, they'll have a nursery, they'll have all these specific things in their tunnels underground. Very cool, very important to their colonial lifestyle. But these settlers said, I don't want these prairie dogs around. I can't prosper with my farm and my prairie like this. So they eradicated them. They started killing them off in huge numbers, poisoning them, shooting them, trapping them, everything they could do to get rid of the prairie dogs. But again, that was the main food of the black-footed ferret. So their population just plummeted. And so, as I said, in the 80s, they thought they were gone. In 1981, they found a very small population, and they pulled them all out of the wild for some captive breeding. So this is one of those situations where if you only have a handful of individuals, if you just let them do their own thing, they wouldn't make it more than a couple more generations. But if you can pull them in, figure out how best to, in the 80s, you didn't have their genes, but you did know who was related to who after one generation. So you can kind of manipulate things so that you're ending up with the least inbred individuals. It's the most genetically viable individuals for the most iterations. And so right now, there's about 650 alive today, which is a good start. But the thing is, that's all from 7 original ferrets.
Talk about a population bottleneck.
Yes, exactly. So they tried to get more of them to reproduce, but only 7 of them were able to reproduce. So that is a huge lack of genetic diversity, huge bottleneck. So this new story is, this is a collaboration with U.S. Fish and Wildlife Service, of course, ViaGen, Revive and Restore, Pets and Equine, the Association of Zoos and Aquariums, and San Diego Zoo Global. They have cloned a black-footed ferret. They used frozen tissue that was stored as part of the frozen zoo that we talk about all the time at the San Diego Zoo. They've been collecting tissue samples for endangered species for a while. And so this was from a ferret that was christened Willa. She died in the mid-1980s. And they were able to use pretty much the exact same method that they used to clone Dolly in 1996, the sheep, very first cloned animal. So they used a surrogate mother of a domestic ferret, the European ferret, with this donor egg. This project actually started back in 2013, so it's been a long time coming. But finally, Elizabeth Ann was born in December.
Elizabeth Ann is so cute.
I know. So good. Yeah.
Elizabeth Ann is just adorable, and now I would like a cloned ferret, please.
Don't. They're bad pets.
I
Go
know.
volunteer at a zoo.
And that
Play with ferrets all
photo,
day.
I didn't know they made sound stages specifically for ferrets.
Oh yeah, for sure.
That's
Yes,
wild.
animal photo shoots are a thing. Yeah, so actually this is the very first clone in the United States of an endangered species. It's been done in other countries, but this is the first time we've done it here. And this is important because she will be able to inject some genetic diversity which is kind of a funny oxymoron, genetic diversity from a clone, right?
Yeah.
But this is going to be some genetic diversity that they'll be able to inject into this gene pool of ferrets.
Right, because it's from an individual that was deceased before this population. It's from somebody else from outside the population of the original 7.
Exactly.
Yes.
Yeah. And so in case you're wondering, I kind of teased it before. The reason you can't have ferrets in, I believe also Oregon and California is because they're escape artists. They're really good at getting out. And if European ferrets got out, they would basically take over the black-footed ferret territory. They could start taking their burrows, they could start taking their food, they'd start taking their resources. So it'd be a concern that this endangered species would get robbed of their niche, essentially.
Yeah, competition.
Yeah, absolutely. So these guys are perfect in the wild, the black-footed ferret, in their own spot. These selective bred individuals slowly being re-released into the wild. Hopefully they won't have a disease come through that affects all of them because they're so genetically related. But for right now, it's doing okay. It's doing good. Cloned endangered species. There you go.
Yay! Happy Elizabeth Ann.
Yes. And from Elizabeth Ann to Saki monkeys in Finland. What? Yes, Saki monkeys in Finland. This is actually a follow-up to a story that I presented last year, which is why I was so excited to bring this story. I was like, this looks familiar. And as I kept reading, so do you remember last year, I talked about monkeys in a zoo that were given a tunnel-shaped device to play different sounds and that they really liked traffic sounds? Do you remember this?
I don't remember that.
Yeah, so they were given the option of rain, music, silence, or traffic, and they preferred traffic. So we had a whole conversation about, is that because they're zoo animals?
Right. Is it because they haven't grown up in the wild? They grew up in a zoo, and is that what? Yeah, okay.
Or is it because it's similar to sounds that they'd hear in the forest? So anyway, so ultimately that was kind of the funny initial story that came out of this. But step 2, what do you think step 2 of this study is? It's TV. Visuals.
They gave them television now?
Yeah, pretty much. No. I mean, really, it's more like a tablet, I suppose. But yeah, it's in a tunnel built of plywood and acrylic, and it has a monitor on the other side. It has, of course, a camera and sensors so that they can figure out what the monkeys are up to. And so it was up to the animals to decide if they wanted to step in. When they go in, the sensor recognizes them there and it turns on the TV. So they could decide, do I want to watch TV right now or do I not want to watch TV right now? And then they would see a video of the week. So during the study, the videos that played were sea life, like fish and jellyfish, wiggly worms, other zoo animals like zebras and deer, abstract art, so like moving abstract colors and stuff, or lush forests. So it was hard for them to identify exactly what their preference was because there was a video of the week every week, so they couldn't pick. But what was clear was they spent most of their time watching worms or underwater scenes. Worms are interesting because they eat worms. But that also those were the things that were available in the middle of the study. So they didn't randomize what was available when. So that means it could have just been when they finally realized how the system worked and they weren't sick of it yet. So difficult to say. It requires further study to figure out what they really like. But what they did find out was that they could really see and discern things on the video because, for example, they tried to lick and eat the worms on the screen. And they even went around to try to see if the worms were behind the screen.
Ooh.
Monkeys.
Yeah. So they could definitely
those monkeys
see
there. I want them.
What was on the screen. The other thing that's interesting here is that compared to groups that had no video content, those groups showed more intense grooming and scratching, which is a sign of stress.
Wait, which group?
The one that didn't have the TV.
The one that didn't have the TV was more stressed?
Well, so they did not test... Yeah. So
Or
this is where I have to be sciencey about it.
Okay.
They did not test hormone levels. They did not pull hairs and test hormones. They did not test blood pressure.
No.
They didn't look at adverse medical impacts or anything like that. So they have like no data here except.
But the monkeys that had TV did more hair combing.
No, the opposite. They did less scratching. Because the scratching is like a bird over preening and pulling out their feathers, So you'll see sometimes if an animal has an adverse behavior like that. This does happen in primates a lot in captivity. If you can't figure out exactly what their issue is, but they'll over scratch and they'll have. It's a lot more complicated than that. But yes, they'll have like a hairless area because they've been scratching a lot. Mm-hmm. And so people spend their entire careers trying to figure out what this animal needs. And it depends from individual to individual. But if this was an option, if this was something in the barrel of potential things to give to an animal who's showing these adverse behaviors, if a TV is one of them that could help them reduce stress and reduce this kind of adverse grooming behavior, that would be a really cool way. And also not that expensive. You just need a wood tunnel with plexiglass and an iPad behind it. So be pretty easy, pretty inexpensive to offer. I will also say when I was a zookeeper, we used to play videos for a lot of the primates when it was really rainy or when they were recovering from a medical procedure and they were shut inside. We would show them videos and they would definitely watch them. Lots of Disney movies, actually.
Oh, that's funny.
I wonder if they get to pick, right?
Yeah, I'm just wondering if the individuals that if they didn't get the videos, the reason that they're itching and scratching is because they are lacking stimulation, or they're jealous because they don't have TVs to watch.
Yeah, I don't know if maybe
it's a dominance thing. Or
I feel like they probably didn't have visual access to the same
other group.
Areas. Yeah. So I would guess that I don't know for sure, but based on what I know about how these things are usually set up, I would guess that they were in a separate enclosure. But yeah, I'm not sure. It's a good question.
Yeah.
I don't know.
Thanks for bringing these two feel-good stories.
Mm-hmm.
Monkeys watching TV.
Aren't we all?
Tonight? Worms.
Oh, that's my favorite.
I hope the monkeys watching their TVs right now are thinking, "Oh, this show, it's great. It's just missing some worms." Yeah. Right.
Here's a worm. Oh.
This is This Week in Science. Thank you all for watching. Thank you so much for joining us for another episode of This Week in Science. If you enjoy TWIS, please consider heading over to our website, twis.org, and clicking on the support us on Patreon button. If you support us on Patreon, you will continue to help us bring this show to you every week. We really do our best to do this over and over again in a fresh and fun way, bringing you new stories and new guests, new content every week. I mean, who wants to watch This Week and Last Week in Science, or This Week and Last Week in, just the same week as always in science? That's not what we do. And it's because of you and your support. So please head over to twis.org, click on that Patreon link, and choose your level of support. $10 and up, and we will thank you by name at the end of the show. Thank you for all of your support. We really cannot do this without you. All right, Justin, you ready to tell us a story? You got some news for us?
Yeah, I got a couple of stories. What makes a stable society? Is it income inequality? Is it a willingness to believe in false information? Could massive deregulation and zero taxes lead to the longevity of a nation state?
I don't think I've ever heard of anything like that.
I don't know if anybody's ever really proposed that, but some people have actually sort of leaned in some of those directions. Studying the journal Current Anthropology by Purdue University. Didn't we have another Purdue story earlier? Anthropologists assembled data on 30 pre-modern societies and conducted a quantitative analysis on the features and durability of these societies to see which ones had good governance. What made a good government? Results showed that societies based on broad, equitable, well-managed tax systems and functioning bureaucracies were more likely to have political institutions that were open to public input and more sensitive to the well-being of the populace, and lasted longer. There weren't very many to choose from, though. From about a quarter of the ones that they had in those 30 sort of fit into the sort of good governance. For more than a century, it's accepted textbook account that democracy was just a Western phenomenon, which, yeah, it kind of was around before that to various levels. But, yeah, it's sort of interesting. It overwhelmingly demonstrates that authoritarian regimes that have broad discretion over a nation's wealth tend to start to use it for more personal and political gain as opposed to the welfare of the nation state as a whole.
Right.
And when you weaken a nation state, it tends to not stick around.
Just skimming off the top and I'll just take a little bit more and a little bit more.
All
and,
of that,
yeah.
all those bureaucracies, all those regulations and red tape and all the taxes you pay, that's the price of being in a civilized society. That's sort of just the way there hasn't been a successful model without taxes and bureaucracy. Anyway.
And the bureaucracy does help to hopefully stop that authoritarian control or use of the funds.
So you could be going anywhere from checks and balances as a thing, or one of the things they sort of pointed to is there's a sort of a positive feedback loop when people are paying taxes and they know what they're being spent on. That they know they're being spent on something that generally benefits them and their society.
Yeah.
Then they're happier in the society. They're more supportive of it. They're more willing to pay taxes to it and work towards the interests of the city-state. And politicians in that system, the bureaucracy side of it is, if you are spending those monies on your nation-state, on trying to improve people's lives, your people will support you. And it was a very positive feedback loop. And I think that's part of what sort of projected the longevity of those tax and good bureaucratic management systems. And then this is the last story that I've got, at least. What makes a healthy diet? We have the 5 food groups. Can you name them?
Isn't that defunct? There's grains.
Okay,
there's dairy,
meats.
Yeah, proteins.
Produce.
Yeah, fruits and vegetables.
Fruits and
vegetables
vegetables.
Are separate. That's 5. You can.
Yeah.
Okay.
And then your yogurt probiotics. Is that where you're going?
Yeah. No. That's dairy. That's definitely in the dairy category.
Well, Is grains? Is that like wheat and rice? That would be
so. Yeah, it's grains or starches. They kind of put grains and starches together.
Okay.
So I guess I don't know, spaghetti noodles, spaghetti pasta.
That's in the grains and starches. Yeah.
What about fats? Is fat something?
That's probably somewhere in the protein foods to dairy, right?
Fats? Okay. No. Well, yeah, it's all included in there. But anyway,
stuff. Yeah. Okay, where do mushrooms fit?
Protein, vegetable.
It's a little. It's sort of. It gives you some of the nutrients that a meat would, and they have schlepped it into the vegetable category because people cook them with vegetables,
right?
Which is not, for a reason to put it into a food category. People cook it with vegetables. So
that's why people think tomatoes are vegetables, right? Goes on your salad. So
no, that's because, I like. Actually, that's very specific. What you just brought up is a very specific thing. The reason it's a vegetable, the reason tomatoes are a vegetable. This was under the Reagan administration.
Oh right,
he
ketchup.
declared it a vegetable so that ketchup could be considered a vegetable in a kid's lunch.
Yeah, I recall that.
So that for the mandatory lunch program that we had to feed all the kids, which is a good thing, you could take out the green beans, I guess, and put ketchup on the menu, and you wouldn't have to have both.
Does a body good.
Yeah.
I would say though, as a longtime vegetarian, little chicken pescatarian eater now, mushrooms have always been a big source of protein. I've always thought of them as an alternative to meat.
Well, think about how many portobello burgers there are.
So many.
Yeah.
But you can do mushroom on pasta. You can do mushrooms are fantastic.
I love mushrooms.
Except for the weird ones that are too chewy, and that's gross. But anyway, that's just me. Other people love them.
So
but go on.
This is a follow-up study from something they were doing last month. They identified reasons to add more mushrooms to the recommended American diet. The slight disclaimer, this is a Mushroom Council funded study.
Oh, jeez.
Okay? But if you think about it, who else is going
disclaimer.
to finance research into the nutritional value of mushrooms other than the Mushroom Council? So they spent like $1.5 million in the last 3 years on
The Fungus
Mushroom
Council.
food research. There's a Mushroom Council, if you didn't know.
I did not know. Now I know.
Huge in the edible mushroom commercial circles. Anyway, the new research is published in Food and Nutrition Research. It examined the addition of mushrooms to the USDA's recommended food plate. So they said if you had this portion in there, which is I think was 84 grams of mushrooms. So about 4 or 5 of those medium to big white mushrooms. Increased several shortfall nutrients, including potassium, as well as vitamin B and minerals, and had minimal impact on calories, sodium, or saturated fat. So it showed here that they had, with that 84 grams of mushrooms, an increase of potassium, 8 to 12%. Copper, 16 to 26%. Selenium, 11 to 23%. Riboflavin, 12 to 18% increase. And niacin, 11 to 26% increase, with just 5 mushrooms a day. And yeah, none of the negatives. Also, oddly, and I don't understand this at all, this I need to have explained to me at some point. Mushrooms exposed to UV light increased vitamin D by 67 to 90%.
Interesting.
Huh?
So they have a process similar to animals in that vitamin D conversion. That sunlight converting cholesterol into vitamin D, probably.
So what you're going to tell me of those blacklight posters of psychedelic colored mushrooms? This is
full of vitamin D.
It's true.
It's true. Good. They fluoresce.
Mm-hmm.
It also found an increase in a lot of other micronutrients that are supposed to be good for you, but the FDA hasn't really said anything about whether they're good for you yet, but it has like some other increasing stuff too. But yeah, so what I thought about this is like how perfect for all of the Nordic and all of Canada, I guess.
For the Pacific Northwest.
Or the Pacific Northwest, where people aren't getting any sun.
Yeah, hello, Eric in Alaska, right?
Here's a way. Get yourself a UV light. They should just pre-UV light them. I don't understand. It seems very specifically like when I don't know if you can still do it once you've bought it from the grocery store.
I've thought about trying to learn to grow my own mushrooms.
There you go. Perfect time. You're in San Francisco. You people are all vitamin D deprived.
Yeah.
There's certain age groups.
Buy a little brick of them, and then you just chop them off every once in a while. Just keep them in a nice place. Not too hot, not too cold. Yeah. I don't think you don't need dirt. They have these bricks, and you just put them in a box, and you leave them in your closet, and they're happy. Yeah. Growing mushrooms is a very fascinating thing.
Okay, so I looked up the food
Kiki knows a guy.
pyramid.
Kiki knows a guy.
Kiki knows a guy.
I know a guy.
So the food pyramid has 6 food groups on it, and the top one is fats, oils, and sweets. But also, all that to say,
it means don't have very much of them. That's why it's at the very top. It's the little portions at the top.
Right, but all that to say, the food pyramid was largely defunct some years ago.
Yeah.
They said it was all a bunch of
There's also a 7-tier pyramid. It's not that it's not all nuts. You can't eat all nuts. That's only one of the 5. See, everybody has their own idea and they're all wrong. But
the main thing is the base of your food pyramid should not be grains. That's really the problem. Is you should not be eating the majority of your food as bread and cereal and rice.
I don't know. I feel like humanity probably for like most of the early civilizations did a lot of that. Did a lot of that.
Yeah, but their bread was very different than ours.
No, it's not. It's the lack of. It's because humans don't move around anymore. We used to walk 50 miles a day in our daily life, and we don't.
Yeah,
We're just lazy. We're just like, but yeah. So my point is,
there is research to suggest that we have always been lazy, that we weren't always on the move. Yes.
You know who did that research? The Council for Couch Manufacturers. That's who did that. See, you always gotta know who did the study.
Oh my goodness. Oh my goodness. Let's move on from couch research into a bit of a seasonal change. Spring is coming, right? Isn't that going to come pretty?
What? Blair, you are the least enthusiastic.
Because what does it matter?
Ever been on this show?
Maybe it has to do with a lack of mu opioid receptors. Or maybe she has too many. Researchers.
Spring looks like fall in San Francisco. It's still foggy. Like it doesn't change.
But I want you to know that researchers decided to look at this question of how opioid receptors in the brain are associated with seasonal changes. Do our receptors in our brain change with the seasons? We already know that there are changes to the brain in terms of... I'll move myself around here. We already know that there are changes to the brain with respect to other neurotransmitters. So dopamine, serotonin, these other neurotransmitters and their receptors also go through seasonal changes. And these researchers are like, well, opioids may be someplace to look as well. Our body produces natural opioids. It's also involved in socio-emotional behaviors. And so they said, let's check this out. Now, interestingly, these researchers are in Finland, in Turku. And so they were looking at changes in day length as the indicator of seasonal change. And in Finland, they have extreme day length changes. So they looked at humans who had been PET scanned, positron emission tomography, and over a period of time. And they looked at their brains to see if there was a change in what are called the mu opioid receptors, or in their acronym in their paper, MOR, M-O-R, the MOR signaling. And while they found a slight change, and it wasn't like a huge link, what they did find enough of a signal in the change in mu opioid receptors, the MORs in the brain, in areas related to socio-emotional cognitive processing. They did find a small link. And so it's very interesting that in addition to these other neurotransmitters, there is now this link with the opioid system and seasonal mood changes, perhaps. They also looked at changes in the brain in the opioid receptors in mice, and they looked at how they changed in their mood over months. And there are a lot of interesting things brought up in this study as evidence, as sources for their ideas moving into this study. Some of them that I found really interesting: the largest number of suicide attempts are in the spring. This is also when the most extreme mu opioid receptor changes have taken place. So you've got the most opioid receptors in this period of time. It's also a period of time in which a lot of opiate not opioid, opiate suicides and overdoses take place. So there is a link, according to them, and the work that they cited, that the summer months are kind of like the sun and everything's happy kind of months. And then the dark, long, dark finish winter changes these opioid levels. People turn to opiate abuse, and then this culminates in a very negative way by springtime after a season of dark depression. But all this aside, what they are very interested in considering is this link between the opiate system, the natural opioid system in the body, and seasonal affective disorders and other mood disorders. And we know that the other neurotransmitters are connected, and now we know that there's a little more evidence that this system is connected to that as well. And why wouldn't it be? Yeah, I thought it was an interesting study to think about. Yeah.
Yeah, I think Norway's too far north.
Yeah, a little bit.
It's one of those places that would probably be really interesting to visit in the summer.
In the summer, yeah. I found the significance of this study originating in a far northern country that experiences 23 and a half hours of darkness in the winter, to be looking at these, for people to be asking these questions, whereas more equatorial areas, people might not be, day length is more steady, right? And where you are on the planet makes a difference, but it's interesting that our brains adapt regardless of where we are because of the differences in day length, and day length can impact our behavior because of those changes to our brain. So I think that's where it really gets really interesting.
So I think it's also interesting because maybe like 10 years ago, if you had talked about getting a sun lamp, people would have looked at you like you were talking about crystals or something. But I guess now there's actually a body of research where you could actually go to a doctor and they could prescribe you a sun lamp. Yeah. That could actually happen, which would be great if you could find a way to help people through seasonal depression. Not that there's anything wrong with pharmaceuticals, but if you could get around that and get through it by getting them a sun lamp, that would be really great. And you could even figure out exactly how much you would need. You would say okay, you have to put it on for this long every day. I don't know. It's very interesting. I would like to see it turned into actual treatment. It'd be neat.
Yeah, and if it could be, it would be amazing. Yeah. And my last story for the night is has a bit more whimsy to it than the previous story, figuring out our own brains. How about figuring out slime mold?
Yes.
Brains. I mean, no, slime mold doesn't have brains, but how do they do things?
They're so cool. I love slime mold.
Slime mold is amazing. It's a basic, it's a biological computer. It's a very basic biological computer, and we're still trying to figure out how it does a lot of the seemingly intelligent feats that it does. And one of those is how it remembers things. So slime mold is known for having memory. And when we talk about that, it's one of the ways it remembers, one of the things it remembers is food sources. That's important to it. Mm-hmm. So if you put a food source in a location and the slime mold is able to detect it and find it, there is somehow a system in place in the slime mold that changes the structure of the slime mold, allowing it to remember where that food source is for a long period of time. Even if another food source might show up someplace else and it might have to grow and move to other places, it could potentially go back to the food source from before. How does that happen? How is it able to calculate that? Well, researchers at the let's see, where are these researchers? These researchers at the Technical University of Munich decided to take a really close look, like under a microscope, at the slime mold. And they've determined that slime mold is made up of these little tubules. And when it is exposed to a food source or something that it might be interested in, what it does is it causes the release of a chemical. And that chemical then spreads through the slime mold to other areas of the slime mold to say, the food is over here, grow in this direction. But, it's a slime mold, so this is a chemical signal. There's no words actually saying this. It's just it's chemical call and response, signal reaction. The trick is that the slime mold, it isn't just branches of mold growing off in a fractally pattern like a tree with its branches. What it is a series of tubes. It really is a series of tubes, and the compound that is the signaling molecule softens the tubes so that it allows the flow of nutrients and also increases the flow of that chemical to other areas in the slime mold. And so the branches, the tubes, get bigger and they grow, and they enable more stuff to flow through them, and they allow more growth to take place. And so it is this structural change that occurs as the result of a chemical signal that leads to this so-called memory of a slime mold. So the structure is indicative of memory.
But you're talking about structure. So maybe I have misunderestimated.
Yeah.
Can we have the picture back up?
Oh,
so
yes.
What you're showing is a petri dish, which at first look appears to be a giant colony that has spread out across of all these tiny single-celled fungi or whatever, this mold creature, like all these single-celled things that spread. But that's not what this is it? Aren't they like weirdly, they're like the largest single-cell creature or something?
They are single-celled, yes.
So
yes, it's a single-celled organism. It's not a bunch of little cells.
No, but what we're looking at isn't a bunch of single-celled creatures, or is it?
No, it is a single-celled organism.
That is one single-celled creature spread out across it. So these things can be like, from what I've heard, like 10 feet. They can stretch out over 10 feet. And there's still this, I'm like not quite understanding this. You understand? That it's not a colony that you're looking at there, but one creature.
Yes.
They're huge.
Physarum polycephalum has no nervous system. It is somewhere between. They say it exists at the crossroads of animals, plants, and fungi. Yeah. It is a link between, I guess, the ancient eukaryotes.
It is a single alien thing.
Yeah, it's a single. It is a single cell. Yeah, and it's made up of these tubes. Bunch of tubes. It's a network of tubes.
Picture that we had up there, and it looks like a sprawling colony that has all these little, like you said, sort of fractally branches going off as it's sort of colonizing the petri dish. But it's not. That's the one creature.
Yes. Single creature.
Insane.
Yeah, so it's not communication between different organisms, between different cells. It's
just it.
It's just it. It's as if a neuron. It's like a neuron sending an action potential or any signal down its length. Except there is no neuron. It's just the cell, the organism. That is what it is. I
remember we reported 100 million years ago on somebody had like hooked up some sort of microchip thing to a slime mold and got it to move in different directions. Could like joystick control it to move around. It's a really strange creature.
Mm-hmm. It's an incredibly strange creature, but they're fascinating, and being able to understand how it is able to do what it does will tell us a lot about about the basics of memory. You know, what is needed for memory? What is needed for behavior? How does, you know, when you break it down to the ultimate simplest form, which this pretty much is, you know, how does it work? And so it's very interesting that what is happening in this particular case is it is a structural change brought about by chemical signals. And the researchers write in their abstract, they combined theoretical model and experimental data to reveal how memory is encoded. A nutrient source locally releases a softening agent that gets transported by the cytoplasmic flows within the tubular network. Tubes receiving a lot of softening agent grow in diameter at the expense of other tubes shrinking. Thereby, the tube's capacity for flow-based transport get permanently upgraded toward the nutrient location, redirecting future decisions and migration. This demonstrates...
This is a logic...
Yeah.
It's a logic machine. It's like the single cell computer.
Yeah, that's what I said. It's like a little basic computer.
It really is. No, I get it. I mean, you said it.
Now you really get it.
It's a computer because it's using logic or something. No, this is that's like a mechanical logic system in place.
Yep.
Wow.
Yeah.
That's amazing.
Yep. This demonstrates that the nutrient location is stored in and retrieved from the network's tube diameter hierarchy.
Mechanical logic gates.
Yeah.
In a I don't even know what to call them. Tube. It's a slime mold.
It's a slime mold. Yeah.
I don't even know how to categorize it.
The blob.
All I have to say is that after this story, I think slime mold is totally tubular, man.
That's only like it's more than 30 years out of date. That it's fine though. It's fine.
It's fine.
Stuff comes around again eventually. Somebody listening to this, you're the Jetsons. We'll totally get that.
Yeah, eventually, one day we will get to the Jetsons. I will have my jetpack. So will you. We'll be happy.
Did you see the new the new post office cars? They look like the Jetsons car.
No, do they?
That would be awesome.
We'll talk about it in the after show.
Oh,
They look so futuristic.
All right, after show it is. I have a letter that I wanted to read from one of our listeners in response to our conversation about fractals a week or so ago. I enjoyed the story about fractals you brought last week. I would like to share something related to it with you. Art is not science, but art can be informed. Art is not science, but art can be informed by science. In particular, music is informed by mathematics, physics, and cognition. Musicologists have long recognized self-similar and recursive structures in music, which are characteristics of fractals. More recently, some composers have intentionally used fractals in their music, usually in digital compositions. I create self-similar recursive patterns of intervals that look like fractals from which I compose. I imagine these fractals to be infinite, but the part that appears in the score is the part that occurs in our hearing range. I used one particular fractal to compose a piece, which is then shared as a YouTube link that I will share with you all on our website if you head to twis.org for the show notes tomorrow after they're posted.
Get to see it now.
It's fractal music, and it goes on. It goes on, and the but the clarinets and the Louis Viquez on clarinets, composed by Paul Lombardi. But I love the idea that music could be that the mathematics of fractals, because music is inherently mathematical, can represent fractals as well. And so I think that in itself is a very, very interesting idea that these repeated patterns and infinite complexity could play off. And so, Paul, thank you so much for sharing your music and sharing your thoughts. Oops, stop screen sharing in my emails. Thank you so much for sharing your thoughts and your composition. I think it is wonderful. Paul is associate professor of music theory and composition at the University of South Dakota, and has been listening to TWIS for a long time.
Nice.
Yeah. If you have any responses to stories that we've had, be sure to email Kirsten at thisweekinscience.com, or there are other emails that we will read off at the end of the show for Justin and Blair and me. And oh my gosh, it is the end of the show.
Oh, what? How'd that happen?
We made it to the end of the show. I want to say thank you to people. Thank you for joining us. Excuse me, I just made myself have a little throat thing.
Okay, so Rod Haglund says, that was Jackson Pollock on clarinet. Do you like a good Jackson Pollock? It's like a scatter plotter of everybody's data with no filters. It's just, it's amazing. And
amazing.
Fun fact, my letter J's, I got
fractals?
I got from the way that my mother does her letter J's, which is this sort of like, it's sort of like, it doesn't look like a regular, it's more triangular, and it just goes down to the bottom. It doesn't have the little loop at the bottom. But I always made them that way because that's how my mom made them. And then I saw one time, and she was an artist who's also a big Jackson Pollock fan. One day I saw his autograph, and I went, oh, she stole his letter J. That's where I got mine from. She absolutely copied it. Her letter J is the J he uses when he signs paintings, which I've used my whole life, thinking I just copied it from my mom, and that was weird. But it turns out she copied it from Jackson Pollock. So it's just it's a fun fact.
Jackson Fly, Justin. Thank you all for listening, for being here, for enjoying the science. I hope you did enjoy the show. I would love to give shout outs right now. Fada, thank you for your help on social media and with show notes. I would love to thank Gord for manning that chat room and heading over to Twitch a couple of times now. I've seen. I'd also like to thank Rachel, our new assistant. Thank you so much for helping out on show things that need to be helped out on. It's just great getting notes on things afterwards to help me manage what we're doing here. And Identity 4. Yes.
We have a new Blair?
Yeah.
No.
No.
Helping out on show.
And thank you to Identity for recording the show. We really could not do this without your recordings. Additionally, I would like to say thank you to our Patreon sponsors. Thank you so much. My camera's in the way. Thank you so much. Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Gaurav Sharma, Shubru, Darwin Hannon, Donald E. Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Runovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Shauna Nina Lamb, John McKee, Greg Riley, Mark Hessenflode, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Melazon, Johnny Gridley, Kevin Railsback, Flying Out, Richard Porter, Christopher Dreyer, Mark Mazzaros, RDM, Greg Briggs, John Atwood, two fabulous thespians, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Mount Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim DePro, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie Stanton, Marjorie and Paul Stanton, Paul Disney, Patrick Pecoraro, Gary S., Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, head over to twis.org and click that Patreon link on next week's show.
We will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and our Facebook channels and from twis.org/live.
We will.
And if you want to listen to us as a podcast, you can search for This Week in Science where podcasts are found. Really cut through all the silly stuff in between. If you enjoyed the show, you can get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can also sign up for our newsletter while you're there.
You can also contact us directly. You can email Kirsten at thisweekinscience.com, Justin at twisminion@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into the tubes of a slime mold.
Oh.
Yeah,
And remembered.
You can also hit us up on the Twitter. We're at TWIS Science, we're at Dr. Kiki, we're at Jackson Fly, we're at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
So I have these new hue lights in here, and my big lights are too strong, but I was trying to like put different colors on. You can kind of see in the background.
That's
right
what I
now,
was doing too.
I've got Soho.
Oh.
I just put on Arctic Aurora.
Arctic Aurora.
Here is, let's see. Where's it?
Make
Oh,
yourself
let's go
big
for-
so we can see your colors.
Tropical Twilight. I don't know if you can see the colors change because my lights are too. You can see the one up at the top, I think, changes just a little bit. Now I'm on Golden Pond. Just changed it to multi. Yeah, you can't really see it. I'm just going to make them bright again. Bright! Be bright!
I have this on my little globe.
Ooh, I like that. That's cool.
It's fun.
Lights are fun.
Look at this bright green. And there's blue,
Ooh, very nice.
purple, orange.
Composition what made you feel like we were part of NPR, Fada? What?
Lello. Ooh, yellow's kind of nice. Maybe I should be using yellow for the show.
It is kind of nice. Here, let me put you there.
Because
that's too blue. It's too white
so
blue.
that's
What's
the
other
white
one?
I've been using. But yeah, maybe yellow.
Yeah, warm it up. I think
Yellow.
the yellow's better. Yellow's
I'll do
better.
yellow next time. Should I do red?
Yeah, Gord, everything is pretty close. But I could.
Or I can strobe. Look
at I was zoomed.
Oh,
Now
dang.
you can see my backdrop. Here. Zoom. Zoom. Oh, with the music, it was very NPR. Yeah, it was a bit.
Oh,
Yeah, the yellow is nice. Everyone agrees.
all right. Well, I'll do yellow next week.
JG, my great granddaddy was a slime mold. Well, at one point, maybe we all evolved from a slime mold. Somebody's grandpappy on down the line was a slime mold somewhere. A fractal slime mold that maybe played music in its tubule appendages like an organ.
It also reminded me of Fantasia, the original Fantasia.
Yeah.
Had a lot of like
Cool.
experimental
Oh,
music
there's
that
Justin.
they've utilized.
Justin came back. There he is. He went away, so. Oh, but you're not there. You gotta unmute yourself, man.
I never left. You just turned the camera off. I've been stuck here in backstage the whole time.
That's not true.
What a liar.
Saw you leave.
So what were you talking about? There was something for the after show that you were going to talk about?
Yes.
What was
no. Oh, Blair had something to talk about, but
she forgot.
the Mars video also.
Oh, the Mars video.
Oh,
That's
the
what
Mars
it was.
video.
I'll
That's
find
what it
that.
was.
Yeah,
That's what
no,
it was.
I was talking about the new mail trucks.
Yeah, that too. Oh, there's all kinds of after show stuff. I mean, we need the mail trucks. We need the
So
the
here,
cell
can
phone
I
from Mars.
screen share?
Yes, screen share some mail trucks, and then I'll find the video.
Okay, so I'm sharing Twitter. I don't think there's
Do
any
you have ad block?
bad language.
What?
Why would there be bad language?
I don't know,
on
just 'cause
a
it's
postal
Twitter.
video?
Oh, right.
That
But
can't
yeah.
be real. No, that's from Up. That's
Yeah.
that's
Like straight out of Pixar.
real.
That's Up.
This is real.
Oh, they're cute.
Oh, that's awesome.
They're
Who
electric.
decided to? Oh, that's awesome.
165,000 electric delivery trucks.
That's great.
Groovy.
That'll be awesome. Okay, where was that video? I know I had
So
it.
everyone's talking about how like they're very cute.
Yes, they are.
Like this person just said, "I love her." And then it looks like my attempt to draw a truck in first grade. True. It looks like it comes from the busy world of Richard Scarry. So there's the lowly worm. And then the other one I liked, it's probably gone now. Oh yeah, I think this is like Johnny Bravo. There's a U.S. mail truck from that show. And then here's Cloudy with a Chance of Meatballs.
Awesome.
And here's the famous car that Homer invented.
Oh yeah. This is Oz brother.
Anyway, I think it's adorable. Look at it go.
Oh,
We
wait.
are starting the straighten up and fly right maneuver.
Ooh.
Yeah, go
Are
ahead.
you ready for 3 minutes and 25 seconds of
Awesomeness.
perseverance?
I don't see it.
Okay, let's watch this. I'm going to share my screen, and then I have to. Oh, never mind. Okay. Ready, set.
We are starting the straighten up and fly right maneuver, where the spacecraft will jettison the entry balance masses in preparation for parachute deploy and to roll over to give the radar a better look at the ground.
Woo, parachute.
Indicate chute deploy.
The navigation has confirmed that the parachute has deployed, and we are seeing significant deceleration in the velocity. Our current velocity is 440 meters per second at an altitude of about 12 kilometers from the surface of Mars.
Heat shield set.
Perseverance has now slowed to subsonic speeds, and the heat shield has been separated. This allows both the radar and the cameras to get their first look at the surface. Current velocity is 145 meters per second at an altitude of about 9.5 kilometers above the
Keep the heat
surface.
shield in the shot. Keep the heat shield in the shot. Oh,
There it is. There it is. Going down there.
My God.
Just falling.
Still a ways. Oh, we lost
Falling
the
to
impact though.
ground.
Oh well.
Yeah. So
Nav
neat.
filter converge. Velocity solution 3.3 meters per second. Altitude 7.4 kilometers.
Now has radar lock on the ground. Current velocity is about 100 meters per second, 6.6 kilometers above the surface of Mars.
Here's that photo that we saw.
Perseverance is continuing to descend on the parachute. We are coming up on the initialization of terrain relative navigation and subsequently the priming of the landing engines. Our current velocity is about 90 meters per second at an altitude of 4.2 kilometers.
LVS valid.
We have confirmation that the lander vision system has produced a valid solution and part of terrain relative navigation.
Priming.
I thought he said salad.
We have
I
priming
heard
of the
that
landing
too.
engines.
I need a Cobb salad. Ordering wow.
Back shell set.
Current
Oh.
velocity is 83
Back
meters
shell
per
came
second
off.
at about 2.6 kilometers from the surface of Mars. We have confirmation
Look
that
at
the
that
back shell has
giant
separated.
cliff.
We are currently performing the divert maneuver. Current velocity is about 75 meters per second at an altitude of about a kilometer off the surface of Mars.
Here in safety Bravo.
We have completed our terrain relative navigation. Current speed is about 30 meters per second, altitude of about 300 meters off the surface of Mars.
I feel like I'm watching
We've
that
started our
Powers of
constant
10 video
velocity
right now.
accordion, which means we are conducting the sky crane, about to conduct the sky crane maneuver.
There goes the sky
Oh, it
crane.
still hadn't
Sky
done
crane
the
maneuver
sky
has
crane. It's
started.
got that much activity on the surface. Wow.
Yeah,
About 20 meters off the surface.
20
Oh,
meters.
there it is. The little jetpack.
Rocks.
Jetpacks are
Mhm.
real, people. They can happen on Mars. There's one right there.
We're getting signals from MRO. Tango Delta.
And there it goes.
Touchdown confirmed. Perseverance safely on the surface of Mars, ready to begin seeking the signs of past life.
Isn't that amazing?
Yeah.
So to be clear, this is the first time we've gotten this view, right?
Yep. First time.
That's a phenomenal.
Cancel.
That's pretty wild.
Cancel.
That's insane. We just watched ourselves land on Mars.
On Mars.
Yep.
Wow.
Yeah, isn't that neat? I know, Gord. It's so cool. Like, I can just watch it all the time. Watch it over and over again. It's amazing.
Like I've said many times, I am perfectly happy here on the moon. You know,
So cool study that it was like an old study that I came across, but researchers are trying to figure out if they can put fish on the moon.
Why?
of course you can. You can put anything on the moon. It's just whether, you know,
Whether it'll
or not
survive.
it
Yeah.
does anything once it's there.
Yeah. So they're French researchers were studying different kinds of fish eggs. I mean, it's not because of caviar and whatnot, but they were studying different kinds of fish eggs to see, like, they were simulating a launch and how much shaking they would be under during a Soyuz launch, and in checking to see how many of those eggs would still be viable and go on to produce fish, because they're thinking that if we have a moon colony at some point, that something like fish would be an easy, if they grow and everything works, because I mean, if they're in water, like,
Well, yeah, if you get enough fish on the moon, then you
Get
can
enough
put whales
fish on-
on the moon. And
Yeah,
then
that
what do you have,
wasn't it.
Blair? If you can put fish on the moon, I'll set it up again. If you can put fish on the moon, then you can put whales on the moon.
I reject.
And then what can you have? What
No.
are you rejecting?
No.
You're the Futurama song, like, jukebox.
We're whalers on the moon. We carry a harpoon, but there ain't no whales, so we tell tall tales and sing a whaling tune.
yay!
Bravo, 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 bravo.
There you go.
Moon tuna. Yes. Yes, but fish on the moon. That is a goal of some
No.
researchers.
Stop.
Maybe. Hi, yawning people. What's going on? And
She
yes,
started
Daniel.
it. She started it, and then it's contagious. and then-
Listen, I haven't slept properly in over a week, okay?
I know. How's your leg?
It's a lot better, but I still can't sleep for more than like a couple hours in a row because I keep waking up in pain. But
Right.
because I go to sleep with like my leg elevated on two pillows with an ice pack on top of my knee, but that doesn't last very long because
Right.
I'm a stomach sleeper normally.
Ooh. See, that would be it right there. I would get terrible sleep just because I'd be on my back.
Yeah. So I get terrible sleep and then I wake up on my side and I'm like, okay, maybe I can figure this out. And I like put a pillow in between my legs and I like put the ice on the side of my knee and I'm like, okay. Then I fall back asleep and then I wake up on my stomach in terrible pain. So I have to like roll back around. But it's getting better.
I'm glad it's getting better.
Yeah, but I'm tired is the point. I'm like, I just,
There you
wow.
are.
As a person who, and I'm aware, I'm planning on starting a family eventually, and it's all going to go out the window, and I'm aware of that. But I just, currently, I sleep, I hit the pillow, I'm asleep in 5 minutes, and I sleep for about 7.5 hours straight.
Amazing.
So this is messing me up.
You're like, what is going on? Well, the fact that you fall asleep quickly is great because you will probably continue to fall asleep very quickly when you are a new parent, if that happens. And yeah, it's just the waking up multiple times during the night. Yeah. The lack of sanity.
Yeah, you hear that?
As you
Sadie?
lose
Don't wake
track
me up.
of time entirely. Nothing makes sense anymore.
Anyway, it's fine. I'll be fine. It won't last forever.
Gaurav, stomach sleeping is fantastic. Just stretch out.
I love it. Well, I'm also like I'm a starfish sleeper also, which like is so usually it's like I'm like this on my stomach, and then this is out, and both legs are akimbo, just completely out.
I like doing that as well. Yep.
Isn't that like a dance move the kids are doing these days?
Taking over the whole bed.
Yeah. Which is one of the reasons that Brian and I feel like get along so well is we only share the bed 30% of the time. So most nights I have the queen bed to myself and I can fully starfish. And I also, I didn't mention, I'm at a diagonal. So my head is like, if you're looking at the bed, my head is the upper right pillow.
Oh, Blair, it gets better with every detail.
So, ma'am, I'm sorry. There isn't a double California king size mattress. That doesn't exist. You could buy
Blair.
2 and put them together, I guess. Yeah, you'd have the gap in the middle, but there isn't.
No. Bunk beds. Bunk bed California king.
I'm sorry, ma'am.
That would be an awesome bunk bed.
Also hasn't been invented yet.
Room of beds.
Dang.
That would be great. Yeah, room of bed. You should just have a room where the whole floor is bed, and you can have like,
fun.
Yeah.
One of the things I always loved
Room
was
of bed.
in the movie Help in the Beatles movie. They had like a crazy house, and they had one of those sunken couches, like circular couches in the floor, and I always thought that was so fun looking.
It does look fun.
Although now that I have an injured knee, I'm like, ooh, that would be really hard to get out of. That's a young person's game, a floor couch.
Yeah. When your knees start to give in your back, then you're like, oh God, I gotta get up. What? I'm just gonna stay down here just a little longer. Room of foam pit. Yes, you could have a foam pit identity for. Yeah, last night I didn't get much sleep either. Don't know what's going on. Don't know what. Is it a full moon? Is that what's happening? No.
Tomorrow, I think. There you go.
That must be it. Must
Your
be it.
circadian rhythms are all met.
They are.
Well, it's already a full
Hot
moon
Rod,
where I am.
what was TMI, Hot
Yeah.
Rod? Was that
On the moon? TMI for your sleeping habits.
Yes, was it the fact that I share a bed with my husband? Was that TMI? You're right. We have twin beds. We have separate twin beds. Like I love Lucy. It's fine. Don't worry about it.
Did that last the whole series, or was that just in the beginning? I feel like they finally got a bed together at some point.
I feel like it was very late in the show.
Yeah, but you're right, they each had their own little twin that they went to bed in.
I think I wouldn't want a twin bed. I like the idea of his and hers king size. I mean,
Bunk beds. That sounds great. I think
the
somebody
bunk beds sound
you just invented something.
fantastic. Yeah.
California King Bunk. That's brilliant.
I hear that many couples, they're very happy not sharing a bed.
Yeah.
It's just like, I can sleep how I want. You sleep how you want. You can. How about we not even have the same room? You can stay up as late as you want. We won't.
Some couples are happier with separate residences,
Yes.
and some couples are even more happy with separate spouses. There's gradations of it, but yeah, people have ways of making relationships work. Sometimes by not ever seeing each other again. It's amazing how many ways there are to get along with somebody.
There are a variety, that is for sure.
I love Lucy. They never had anything but the twin beds.
Yeah.
Okay.
No,
So
they
it was
wouldn't
the-
be
it was
allowed
the
on
censorship
television.
thing.
Rafe
Yeah.
Awkward Primate says, I sleep on the couch every night. There was one point when I had the best couch, which is gone now because mice attacked it one time.
Oh,
and killed it.
no.
But I had the best
Bummer.
couch. And it got to the point where I had moved into a new place and I was like, I don't remember the last time I slept in the bed, and it takes up a tremendous amount of room. And I got rid of the bed. I just got rid of it. I'm like, the couch is perfect. I like sleeping in it. I prefer the couch is the best night's sleep I get. That's it. And I just had that couch for some time.
Wow.
Yeah.
Huh. Did you move the couch into the bedroom?
No. But the bedroom became then the just a TWIS studio. Yeah. It's just like I just had desks in the bedroom.
So, do you think it was the couch, or do you think it was the fact that you like were doing other things and then would just succumb when sleep took you?
No. I would if I could. I'd toss and turn in its nice queen size bed. It was tossing and turning.
Yeah.
I'd get up, go lie down on the couch, and be out like that.
Wow.
It was just a better.
That just sounded like you had a bad bed.
The bed was fine. I haven't met other beds that I'm like, ooh, this is what I was missing.
Really? You've never
This is
slept
what
on a bed in like a hotel or anything and been like, oh shit, can I take this to go?
No, actually, every hotel bed I've ever been in, I'm like, this is awful. Hotel beds are the worst.
Really? I've slept on a couple that were like, if I could have stuffed it in my suitcase, I would have. It was so good. But I think they were pretty fancy hotel rooms. Like,
I mean, wasn't that the whole thing with some of the big fancy hotels where they're like, "You love our beds, now buy them," and that turned into a whole commercial.
oh
like,
Yeah. The
Thing for them.
problem is you bought beds that they had for a long time, so they always had stains on them.
No.
Like how many thousands of different people had slept in those beds? It was awful.
No.
That was a bad plan.
That was not the plan. It's not what you did. You're not buying used mattresses from a hotel.
Gotta get rid of them
No.
somehow.
no.
No, thank you.
Where do you think all those discount mattresses come from?
Oh, Justin.
With recycled materials? You know what recycling means? They took the stains out. Most of them. That's how they're recycled. Okay. I think we've come to that point. Blair, you've been yawning all night. I think it's time for you to say goodnight, Blair.
Okay. Good night, Blair.
Okay.
Say goodnight, Justin.
Good night, Justin.
Oh no, I forgot to remind everybody. Mark your calendars, April 17th, 4 to 6 p.m. for the This Week in Science Daily Tech News Show Super Show Science Tech Extravaganza.
Didn't you make a joke that wasn't the name of our show earlier? We were like, it's This Week in Science, not this Daily Tech News web night and last week's show and every day the same day in science.
I kind of.
But we still forgot to plug it
I wrote.
during
Yeah,
the show.
for the show. For show. Yeah,
You know where we should put the reminder?
on the website.
Show notes.
In the show notes.
Website would be good and show notes.
Yes.
Yeah.
Well, you
I like
see,
this.
next week is a new month, though. Yeah. I'll have to remember to transfer.
To put it in the new. We need to do show notes. So we need new show notes. We need new notes for the show notes. We need to shout out those notes in the show. And it's
Justin,
fine.
why don't you write a disclaimer about it next week?
The faux show notes.
Yeah, I will. But it's going to be the week after it happened. Sometimes you make notes, you leave a note, and then you set something down on top of the notes. And then a week after the notes were really prescient, you find the note again. And it's too late. Can't even make a note to remind you that the note needs to be
Noted.
accomplished. 'Cause
Noted.
it's no longer.
Take note of the note.
Good
night,
Kiki.
Good night, everyone. Good night, everyone. Thank you for watching, and we do hope that you will come back again next week. Tell your friends about TWIS. Make sure you're subscribed. Click that like button down below if you're on Facebook or YouTube. Just Twitch have likes? Do the likes. I don't know. Tell people. We like you. Thank you for joining us. We'll see you next week. I'm going to end this thing. Bye.